welcome to this class on neuroscience of human movement. In today's class, we will be discussing cerebellum, and it will be a summary of the discussion, the previous discussion. So, for the past 11 videos, we have been discussing cerebellum, its anatomy, its functions, pathways, and what could happen in case of lesions, etc. In this class, we will summarize. So, some details may be missing. Please do check back the original video to understand the details. Here the idea is to provide just a relatively brief summary of the topics. So, let us remind ourselves of the situation with the movement system or the neural control of movement, the system that is responsible for neural control of movement. We said that movements are generated through skeletal muscles, and skeletal muscles are controlled by alpha motor neuronal pool in the spinal cord. So, that is the spinal cord, the dotted box is the spinal cord, and these alpha motor neuronal pool are controlled by local neuronal circuits. There is sensory information that also comes from the muscle, for example, and from other sources, of course. And we have discussed the skeletal muscle physiology of that and how movements are generated, how muscles are responsible for movement generation. We have discussed the connectivity within the spinal cord, excitation and inhibition within the spinal cord, how alpha motor neurons are getting excited, how they may be inhibited, how sensory information causes alpha motor neurons to fire or get inhibited, as the case may be. And we have discussed how these spinal motor neuron circuitry and its function is controlled to a large extent by the motor neurons or the motor areas of the cortex. This is primary motor cortex or simply M1, or supplementary motor area, and then other motor areas. We have discussed the other areas. This is the pre-supplementary motor area and dorsal premotor area, ventral premotor area, pre-dorsal premotor area, etc. We have discussed all these cases in the previous classes, and we have discussed so far in the last 11 lectures, we have discussed the role of cerebellum in modulating movement. Importantly, please note that the cerebellum receives input from the spinal cord, that is a one way arrow, and it communicates not directly with the spinal cord, but rather with the motor cortex and the brain stem. So, it does not directly control movements, whereas it modulates movements or it regulates movements. So, in other words, it does not communicate with the spinal cord. The cerebellum has no direct projections to the spinal cord, although the cerebellum has projections through the vestibulospinal tract, rubrospinal tract, reticulospinal tract, and through the cortex, as in it projects to the ventrolateral thalamus, from the thalamus to the cortex, and from cortex to through the corticospinal tract. Right. So, we have discussed these topics, we have discussed cerebellum. Somatosensory and the sensory areas we will not discuss as part of this course, we have mentioned this previously. So, the next topic of discussion will be basal ganglia. So, let us remind ourselves of what we learnt in the past 11 lectures. Okay. So, this is a picture that shows the anatomy of the cerebellum. There is the anterior lobe and there is the posterior lobe. So, that is the posterior lobe, relatively big lobe is posterior lobe. These two are separated by the primary fissure, and there is a fissure that separates the posterior lobe from what is called as the flocculonodular lobe. To remind ourselves, the flocculonodular lobe is also called as the vestibulocerebellum or the archicerebellum. This is the old cerebellum that is found even in fishes, right. So, evolutionarily this is the earliest precursor to the cerebellum. further development led to what is called as the spinocerebellum. That spinocerebellum is basically the vermis and the intermediate zone or the paravermal area. This is called as the spinocerebellum. This is called as a vestibulocerebellum. Vestibulocerebellum or the archicerebellum is called as the spinocerebellum or the paleo, relatively old but not too old, paleocerebellum. The lateral areas are well developed in advanced species, in more developed species such as primates and humans, that is called as the cerebrocerebellum, also called as pontocerebellum, also called as the neocerebellum. It is relatively new, so it is called as neocerebellum. Their functions are different, the areas to which they project are different, the inputs, the areas from which they receive inputs are different. So, we have discussed these cases separately in vestibulocerebellum for a couple of classes, or maybe the spinocerebellum for 1 or 2 classes and the cerebrocerebellum for 1 or 2 classes. And the lesions that happen to these regions also cause characteristic differences. So, there will be a difference depending on the region of the lesion, the response of the system of the organism will be different, etc. So, the flocculonodular lobe, what is not mentioned is the flocculonodular lobe is separated from the posterior lobe through the posterior fissure, and the anterior lobe and posterior lobe are separated by the primary fissure. Okay. Also in the discussion of the microcircuitry, we said that there are these climbing fibers that originate in the inferior olive. So, there are 2 major sources of inputs. One is the climbing fiber exclusively originating from the inferior olive. If a fiber originates from the inferior olive and projects to the cerebellum, it is called as a climbing fiber. There is a reason why this is called as a climbing fiber, because this climbs through the tree like structure of the Purkinje cell and makes multiple synapses with its dendrites, and all the other inputs that originate from all other sources such as pons. What is the source of information to the pons? Pons receives input from the cortex and then projects. So, pons acts as the relay center by receiving input from the cortex and then projecting to the cerebellum, and these inputs are coming through what are called as mossy fibers and spinal cord, vestibulonuclei. Any source that is not the inferior olive, if it enters, if it gives an input to the cerebellum, it is called as that fiber is called as a mossy fiber. Okay. The way these two systems work is fundamentally different. So, we will discuss that very briefly. We have had a relatively long discussion of this topic in previous classes. So, but I will try to remind you of this. It turns out that the Purkinje cell is 2 dimensional. So, it is thick in one dimension and relatively thin in the other dimension, right. So, in the dimension in which it is thin, it receives inputs from millions of parallel fibers. This has not been discussed previously in this class, as in this has not been discussed previously in today's class. The mossy fibers make synapses with what are called as granule cells, whose axons go on to become parallel fibers. So, granule cells. So, granule cells receive input from mossy fibers and their axons go on to become parallel fibers, and each Purkinje cell, which is an inhibitory neuron, receives input from hundreds of thousands of parallel fibers, right. So, let us remember that there are about 100 billion granule cells. So, there is a great divergence of information from the mossy fiber system. There is a smaller number of mossy fibers, and so each mossy fiber makes synapses with thousands of granule cells, whereas each granule cell receives information only from a few mossy fibers. Also note that the climbing fiber relationship with Purkinje cells is also special. The climbing fibers, each climbing fiber projects to about 1 to 10 Purkinje cell, but a given Purkinje cell receives input from one and only one climbing fiber. The way these two function and the balance of excitation between these two systems probably form a crucial role in the function of the cerebellum as a whole. So, what you see here once again, let us remind ourselves of the situation. So, the layer where the dendrites of the Purkinje cell is located and other neurons and the axons of the parallel fibers are located, this is called as the molecular layer. The layer where the Purkinje cell bodies are located is called as the Purkinje cell layer, and the layer where the granule cell bodies are located or just below the Purkinje cell is called as the granule cell layer, right. And so, mossy fibers innervate granule cells and they go on to become parallel fibers, and they innervate the Purkinje cell perpendicular to each other. We showed this example of cables passing through sheets of paper. So, this sheet of paper is it is 2 dimensional, it is very it has area in this dimension, but in this dimension it is relatively thin and the parallel fibers go through this. So, this is the Purkinje cell, as in that is the Purkinje cell, and the parallel fibers are perpendicular to the Purkinje cell. This we discussed in the previous class. So, because of this reason the parallel fibers are able to innervate a number of Purkinje cells, and each Purkinje cell receives input from a whole bunch of parallel fibers, hundreds of thousands of parallel fibers. The number of dendrites in Purkinje fibers can be as high as 250,000, right? Relatively large numbers these. So, this important feature of the cerebellum or this microcircuitry of the cerebellum gets repeated multiple times, thousands of times. So, this function of the cerebellum is probably predictable by function of this simple microcircuit. So, if we are able to understand how this microcircuit works, to some extent, at least to some extent, we should be in a position to understand how the cerebellum works. The details of how exactly the cerebellum works, all its functions and its abilities continue to remain a mystery, but to some extent we are able to uncover that mystery by understanding of the neurophysiology of how this microcircuit works. Okay. And we discussed the recurrent loops that the cerebellum has with cerebral cortex. So, cerebral cortex sends neurons or sends information to the pons, and pons acts as the, so that is pons or pontine nucleus, pons acts as relay center and it sends information to lateral cerebellum. Lateral cerebellum means the cerebrocerebellum, and without the detail of how exactly this is happening, from the lateral cerebellum basically there is projection back to the dentate nucleus, that is not discussed. So, there is projection to the dentate nucleus, which is one of the deep nuclei of the cerebellum. So, there are 4 nuclei of the cerebellum, these are dentate nucleus, fastigial nucleus, globose and emboliform nuclei, right, and the globose and emboliform together are called as interposed nuclei sometimes, right. So, the dentate nucleus projects to the thalamus, and from the thalamus the information goes back to the cerebral cortex. Most of the time this information goes back to the motor area of the cerebral cortex, thus modulating the motor function, right. And let us remember that if the dentate nucleus is cooled or inactivated or deactivated, then what happens is that information does not go from the cerebellum to the thalamus and from the thalamus to the cerebral cortex. But this does not prevent movements from happening, but rather this delays the movement and reduces coordination. We saw this in one of the previous classes where we showed that cooling of the dentate nucleus produces symptoms typical of the cerebellar disorders, right. So, there will be tremors, there is overshoot and undershoot, etc. Also another recurrent loop is with the inferior olive. It turns out that the cerebellum receives input from the climbing fibers. Actually, it is a very strong input, because the climbing fibers produce what is called as a complex spike. This is because of protracted or prolonged calcium conductance in the climbing fiber Purkinje cell synapse, right. So, this has a strong effect on the Purkinje cell, and the Purkinje cell inhibits the deep cerebellar nuclei, right. It turns out that what is also true is that the deep cerebellar nuclei can have inhibitory effect. Actually, there is an inhibitory sign that is there. So, have an inhibitory effect on the inferior olive. A special feature of this inferior olive neurons is that these neurons or neurons within the inferior olive can communicate with each other electrotonically through gap junctions. It is a very special feature, not present in many other cases, right. So, in a way the cerebellum controls its inputs to some extent, at least it regulates what sort of input it receives and to what extent these inputs are influential to the cerebellum itself. So, cerebellum receives inputs, it also regulates its inputs, or it regulates at least one of its strong inputs, which is the climbing fiber through the inferior olive. So, this is the recurrent loop that the cerebellum has with the inferior olive. This is the previous case that we saw where the cerebellum having a recurrent loop with the cerebral cortex. Another recurrent loop involves loops that exist within the cerebellum. In this case, it turns out that the mossy parallel fiber system excites an interneuron, a type of interneuron, inhibitory interneuron called as Golgi cells. These Golgi cells in turn inhibit granule cells. Actually, it is the granule cell or the parallel fiber that excites the Golgi cells. They in turn inhibit the granule cells. Let us remind ourselves this: granule cells are very large in number. We have 100 billion granule cells. If all of these are active simultaneously, it is not clear what is the information that is being communicated. So, to ensure that we get the information that we want while avoiding unwanted information, or in other words noise, it is important to keep a large number of these granule cells silent. In that sense, Golgi cells, it is believed, or an interesting hypothesis is that the Golgi cells are keeping a large number of granule cells silent, so that whenever the granule cells are active, the information that is communicated by them is in some sense more informative, or in some sense this is helping the cerebellum code information in some form of sparse code, what could technically be called in engineering parlance as sparse code. Yes, probably. This is an interesting hypothesis, this continues to be researched actively. And the pathways within the vestibulocerebellum. So, it turns out that the information from the vestibular labyrinth goes to the vestibular nucleus and to the vestibulocerebellum, which is the floccular nodular lobe here, and from there it projects back to the vestibular nuclei, and from there to the vestibulospinal tract. An interesting point to note here is that information from here does not go to any of these. These are the deep cerebellar nuclei, these are the major output structures of the cerebellum. The vestibular nuclei are considered as both input also as the major output system. So, in some sense the vestibular nucleus is comparable to the deep nuclei in the sense that it is acting as an output nucleus of the cerebellum. Okay. and within the spinocerebellar case there are a few pathways. So, here you have the interposed nuclei, these are the interposed nuclei, we said this is the globose and emboliform or the interposed nuclei. So, information coming in from the vermal and the paravermal area, right. So, they project to the red nucleus and from the red nucleus to the lateral descending tract. Important to note here is that the cerebellum controls movements or controls parts of the body that are on the same side. So, this may happen in multiple ways. This may happen in a way where the projection from the cerebellum is to the same side of the body, it is not going to be direct, the indirect projection is going to be on the same side of the body, or it is possible that there can be two crossovers like it happens in this case. Okay. So, it is crucial to note whether there are two crossovers or not, but regardless of whether there are two crossovers or whether there is no crossover, it is important to realize that cerebellum controls body parts that are on the same side or ipsilaterally, whereas cerebral cortex, whereas the motor cortex controls parts of the body that are on the opposite side or the contralateral side. So, within the vermal area, for example, there is projection to the fastigial nucleus and from the fastigial nucleus to the vestibular nuclei through the reticular formation, and from the reticular formation through the medial descending tract, also called as a reticulospinal tract. This is called the red nucleus pathway is also called as the rubrospinal tract, and from the vestibular nucleus there is output that is called as a vestibulospinal tract. Okay. There are multiple inputs that the spinocerebellum receives. What is the spinocerebellum? Spinocerebellum is this intermediate zone, this is the vermis, that is vermis, and this is the paravermal area or the intermediate zone, right. The most medial and the neighboring areas are called as the spinocerebellum, is it not? It receives inputs from multiple sources, but all of them are coming from the spinal cord mainly. So, what are the pathways? These are called as the spinocerebellar and the cuneo-cerebellar pathways, and spinal cord projects to the reticular formation and multiple nuclei within the reticular formation such as the lateral reticular nucleus, such as reticularis tegmenti and pontis paramedian reticular nucleus, for example. From there is projection to the spinocerebellum. So, that is one, there is more actually, and the spinocerebellum projects back to the reticular formation, and the reticular formation controls the spinal cord via the reticulospinal tract, and spinocerebellum also projects to the red nucleus and through to the rubrospinal tract controls the spinal cord. So, note here from the spinocerebellum there is a direct connection, this is not there. There is a direct projection to the spinal cord, but rather it is only through reticular formation and red nucleus in the case of spinocerebellum. In the case of cerebrocerebellum, it is going to be through thalamus and cortex, that is in the next few slides. Okay. So, there are multiple cases. So, for example, so there is, it also projects in some cases to the ventrolateral thalamus and cerebral cortex through the corticospinal tract. So, information is coming in from multiple sources, from spinocerebellar, cuneo-cerebellar tract, from the reticular formation, and it is projection, the outputs are projected to reticular formation, red nucleus and to thalamus cortex, and the outputs are finally implemented, the final execution is performed through reticulospinal tract, rubrospinal tract, corticospinal tract, etc. Okay. And for the cerebrocerebellum case, information comes in from the cerebral cortex to pons, and from pons to through the lateral areas, or this is the cerebrocerebellum, through the lateral hemispheres, and projects to the dentate nucleus, that is the big nucleus, right. Through the dentate nucleus, this is relatively well developed in advanced species such as humans, right, and this is mainly through the middle cerebellar peduncle. So, middle cerebellar peduncle is exclusively dedicated for communication with the cortex, right, and from there to the ventrolateral thalamus through the superior peduncle, and from the ventrolateral thalamus back to the cerebral cortex. This is the recurrent loop that we saw in one of the previous slides, right. So, a very brief overview of the cerebellar peduncles here. So, parts of the, this is the sub part of the cerebellum or area of the cerebellum. If it is a cerebrocerebellum, it receives input from the cerebral cortex via the pons and it outputs through the dentate nucleus, and the input peduncle is mainly middle peduncle and the output peduncle is mainly superior peduncle. If it is the spinocerebellum, it receives input from the spinal cord from multiple sources through the cuneo-cerebellar tract, through the spinocerebellar tract. Actually there are two divisions of that, the dorsal spinocerebellar tract, the ventral spinocerebellar tract, etc., and the cuneo-cerebellar tract. And the deep nuclei involved are usually the interposed nuclei. The input peduncles are not just one, in this case it may be either superior or inferior, the output peduncle is mainly the superior peduncle. And if it is a vestibulocerebellum, then it receives input mainly from the vestibular nucleus, and the deep nuclei involved are fastigial nuclei, sometimes it is the vestibular nuclei, and the input peduncle is mainly inferior, the output peduncle is also inferior, right. So, one could see that in this case, by looking at the peduncles, you could say that the middle peduncle is mainly an input peduncle, the superior peduncle is mainly an output peduncle mostly. The inferior peduncle is a relatively complicated structure, it is a smallest, but it is relatively complicated, that has both input and output projections. Okay. And we discussed the case of the role of cerebellum in planning and regulation of movements. So, feedback about movements arrive at the cerebellum through multiple sources. It also goes to other sources such as motor cortex. For example, the movement is being made, motor cortex receives input or feedback via the association cortices, that is shown here, and cerebellum receives information about the planned movement. How does this receive the information? Via the dorsal and the ventral spinocerebellar tract. What it receives, what is the kind of information that it receives? Proprioceptive information and what is called as efference copy. So, cerebellum knows the current state, cerebellum knows what is the state that is going to happen, what is the current command, and it knows the results. So, it knows the current state, it knows the command, it knows the intended state, it knows the result of the command. How does it know the result of the command? The result of the command is known by proprioceptive information. I have given this command, I want to make that movement, but whether or not I have made that movement can be understood by comparing the intended state and the result. If these two are the same, then there is no need to make any correction. If these two are not the same, that is a movement error, that must be corrected. So, there are multiple sources of information. How does the cerebellum know the intended state? That is coming from other sources, from motor areas of the cortex and brainstem that project to the cerebellum. This is what the intended state is. How does it know the command? That is the efference copy. Command is the efference copy. How does it know the current state? Once again through the proprioceptive information. So, multiple sources of information are integrated in the cerebellum and it functions mainly as a error correcting engine, right. So, and it does not perform this error correction directly, it always projects to the cortex via the thalamus or to brainstem via the other sources such as red nucleus reticular formation, right. It projects to other motor areas and these motor areas project to the spinal cord via the vestibulospinal tract or rubrospinal tract, reticulospinal tract or corticospinal tract. So, In other words, cerebellum gets this information on multiple factors and helps to regulate or modulate movements. Okay. So, it is also believed that there are other functions of the cerebellum such as cognitive functions, but before that there are functions such as balance and eye movements such as vestibulo-ocular reflex. We discussed this case in one of the previous classes, and it is believed that parallel fibers perform a crucial role in motor coordination. Parallel fibers project perpendicularly to the Purkinje cells. Because they innervate Purkinje cells at multiple levels, it is possible that these Purkinje cells which project to the deep nuclei in a somatotopic manner. So, that means if different Purkinje cells are activated, that means that there could be multi muscle coordination or multi effector coordination, in other words motor coordination. And modulation of eye movements by area in the medial cerebellar cortex. So, the vermal area, and the other contributions of parallel fibers in motor function, and then most importantly motor learning. What we said about motor learning was that the climbing fiber acts as a teaching or instructing signal by simultaneously activating the error causing by simultaneous activation of the Purkinje cell along with an error. So, whenever an error is happening, a set of parallel fibers have fired, right, are firing at that time. At around the same time, if the climbing fiber is also firing, this causes what is called as a long term depression in the Purkinje cell parallel fiber synapse. So, in other words, it is telling the Purkinje cell, or it is informing, it is communicating to this Purkinje cell that these parallel fibers are causing errors. Repeated effects or repetitive actions of this, for example, multiple times when the same thing happens, as in the same error is happening, this climbing fiber is again informing, causes this long term depression to become, or for the climbing fiber to continuously activate the Purkinje cell. That way it is reducing the possibility that these parallel fibers are going to be responded to in future. So, that way there is an error correction that happens in relatively long term. This could last 2 hours, 2 days. Okay. So, with this we come to the end of this class. Thank you very much for your attention, and we will continue our discussion on basal ganglia in future classes. So, thank you very much.